So I wanted to put my thank you slide first, mostly just so I don't forget. Most of the new work that I'm going to talk about today was done by a very talented graduate student at UC Davis, Kelly Gutierrez, whose advisor is Bob Guy, who almost everybody in this room knows, my very close collaborator at UC Davis, who pretty much is the only thing I miss from Davis. But it's a really big thing, so it's a big loss for me being at Smith. But I'm obviously really happy to have relocated, and for lots of personal reasons that I'm happy to talk about offline. But I did have a nice meeting with him yesterday. I do meet with him quite regularly, and he says hi to everybody and happy birthday. So I also want to acknowledge the work I've been doing with Smith undergraduates and my very close experimental collaborator at UPenn. So my motivation in general is to think about how, so we're leaving the inertial realm, okay? Don't worry. For those of you who are uncomfortable in the last talk, we're back to zero Reynolds number. So the motion of cilia and flagella is, of course, very important to human health. And in particular, something that I'm very motivated by is how sperm navigate in the very complicated environments they live in. And cilia transport and mucus in our lungs is, of course, very important. So what these objects have in common is the thin hair-like structures, flagella, that move at these really small scales. It's kind of funny. This is an aside. I was giving a talk at Smith, and it was faculty talk, and there's a woman who does sperm, she's a retired biology professor who's been working on sperm for a really long time. And she said she hates the word fertilization. She doesn't want to talk about how sperm move because it's all about the woman, and the woman bring in the sperm, and it's not about the men. And it was hilarious because, there I am trying to learn something. And I was like, we're at a women's college. Like, it's really different here. It's not about the men, guys. Okay. So it's not about the sperm. All right. But I study the sperm. So I'm like, okay, I don't know anything. I'm not a biologist. So, all right. Anyway, that was an aside. Forgot that this was being recorded for posterity. So microorganisms move in complex environments at very small scales. We all know this. And I'm going to focus on fluids with elasticity. So I don't typically use regularized Stokeslets in my work because I use models that include nonlinearities from elastic structures. However, this is a special talk for Ricardo, and there will be regularized Stokeslets at the end. So wait for it. Of course, we know, and something that I think about a lot is how gates change as fluids change, because this can be very functionally important. Certain sperm swim in mucus, certain sperm swim in water. What's going on? Why would gate changes happen? It's way more complicated than just what's the fluid they're swimming in. And in particular, something that I'm starting to think about is how the flagella themselves work. But what I spent a long time thinking about was nonlinear interactions of the fluid and the body and the resultant gait of the organism. So to kind of put a little bit of a better picture on all of this, I have a few examples. And so if you look at images of sperm motility from the early 90s and the group of Suarez, they were the first to really see how kinematics depend on the activity state of the organism on the sperm, but also the fluid viscosity. And so you'll see, there's a point. Let's see. That probably has to be turned on. Maybe not. We'll just skip that. You see. Then actually, so here are some nematodes. So we can remember back to the previous slide. These are small nematodes. You can still think of zero Reynolds number. But work in the both nematodes and then the Chlamydomonas algae cell, people were seeing that organisms both slow down when they're swimming in higher viscosity fluids, as well as change their gait. And in particular, if you'll see on the bottom in the middle, you have on the left, you have this clammy stroke, which is swimming in a Newtonian fluid and then in a viscoelastic fluid. And there are significant gait changes, but even more significantly, and something I'll point out today in particular, is there's significant frequency changes also. And so if you look at the chemographs and you see these waves traveling on on the flagella, you see, like 4 stripes on the left and like 7 stripes on the right, indicating that there's been a significant increase in the frequency as well. And that's not something I had thought about until more recently. And I'm not going to talk about the frequency too much today, but just to say that that's an important, it's complicated in modeling to think about the frequency. So we'll get to that. And then more recently, the group of Jeff Guasto, he analyzed a whole bunch of different sperm in a bunch of different environments and made some really interesting observations about the sperm flagellar shape and its dependence on the environment. And in particular, what they observed was that the shapes, if you measure distances in shapes, which is quite complicated and you think about how you want to do that, but if you look at the different shapes, the shapes were more organized by the fluid than they were by the species. And so you ended up with some vertebrate species being more different if the fluid that they fertilize in was water versus mucus. Okay, and so the separations due to the fluid was more important. And so that, of course, leads to the question of like how do these gates arise? How much is evolution? What is really driving those motions? So I'm not going to spend too much time about this. Of course, we know when we think about movement through a fluid, we have balance of momentum, we have the Reynolds number, we set the Reynolds number to 0. And but this is like my favorite slide that I've made recently, where I like think about how you move in water versus moving on the sand. I don't really like that picture on the left. That guy kind of bothers me, but I tried to make AI make a better picture for me and I failed miserably. It just shows I'm too old. Like I can't use AI to make pictures. My kids need to do it for me. I'll ask them next time. But anyway, if you stop moving your body, you stop, right? That's like swimming in sand. And so you need alternative swimming strategies. And this is, a famous pictures from a famous talk by Purcell, where we talk about how you need to move your body by some sort of non-time reversible motion, right? And so you have to pass a traveling wave down your body, or you have to have these long flagella. And flagella and cilia are tail-like or hair-like filaments. And they propel the organism by generating beats. And in particular, when you're at the eukaryotic type of flagella of organism, then you have actual motors along the flagella that cause them to bend. And beats are a result of coupled interactions from the surrounding fluid, elastic forces of the bending filament, and motor activity. And that is a part of the model that a lot of people have been working on and trying to develop models with motors in them for a long time. But none of those models are really ever put into viscoelastic fluid. So that's yet sort of like a cool new modeling knob to turn, right? If you think about the motors and you think about fluid elasticity and how they interact. So kind of that's where we're going in our big research project of trying to add motors to our models. We're not there yet, but we're so much closer than we were. So it's kind of exciting. So let's just really quickly go back to that goo. Viscoelastic fluid has, we can think of it as a solvent plus something inside. I think of like rubber bands. They flow like water, but they can remember their shape for some characteristic time scale. I was going to take this out of my slide, but then Tom Beale said it sounded like it was cool, so I put it back in. So this is a movie I made back during COVID when I realized I needed to explain different properties to people, and it was a lot easier with like a picture. So I'm gonna talk you through what we've seen in a minute, but we're kind of looking at some familiar materials and I'm playing with them. You can guess what they are. I mean, it's kind of obvious, but just in case. Silly putty, right? So that's actually my hair gel. So all of these were taken right from my bathroom. And something notable about this particular picture is the hair gel doesn't really fall unless you wait long enough. And there's obviously some toothpaste, and I want you to observe kind of the way it's coming out. You all like to cook and you like to make cake or play with mucus, for example. kind of like mucus substance, whites. Just a couple more. This might be the last one. What do you think? That's cornstarch and water, yes. Your favorite. I could probably try to make this video better, but you know, I'd need like a hand model. I hate my hands. Ever since I had a teacher in the 10th grade telling me I had the most manly looking hands, it was really weird. Traumatizing. All right, so what are some of the properties we saw? Well, we saw water at the very beginning while I was rambling. We saw a rubber ball. And these are kind of the opposites, right? We've got like a classic viscous fluid, water, and the viscosity is basically the flow rate. The rubber ball is a classic elastic solid. And the thing about the rubber ball is when you push on it, it comes back to its shape. It remembers its original shape forever. A memory foam pillow. If you remember, I put my hand on a pillow. That was my pillow. I love that pillow. And it remembers the shape of your hand for a little bit of time, and then that goes away. And that's the finite memory time that I talk about. And then some of the familiar ones. We've got shear thinning, which is an important property which happens for certain materials where it allows for easy flowing outside the edges. So like toothpaste is shear thinning so that the toothpaste actually comes out in a nice rectangular shape. Then we've got egg. Egg is probably, the egg white's probably the closest to the viscoelastic fluid of anything that I was playing with. So, all right, so we're going to use the Maxwell model for viscoelastic fluid, which actually Erin talked about briefly. And this is a model that comes when you think about, and it's a really good thought, you know, like just a mental model to have when you're thinking about viscoelastic fluids, where you imagine a linear arrangement of a spring in a dash pot. And so you have the spring has some elastic modulus. You can think of it linear Hooke's law. The viscosity comes from what I was saying before, that the viscosity is proportional to the flow rate. And so if you take this model and you try to assume that the stress is fixed, so tau P equals tau S, and the strain is additive, so epsilon, the total strain, is the sum of the solvent and the polymer strain, and you do some like little manipulation, you can derive the Maxwell model, which is down there at the bottom, which is this ODE, which tells you that the stress has a time history of, remembers that deformation on some sort of time scale. And so from this, you can define the relaxation time, which is this mu P over E, and you end up with a Maxwell model that looks something like that. And if you are just willing to believe the mathematicians and many people who've done tons of work on this, if you think about the time derivative of a tensor along streamlines, you don't want to do just tau dot, which is time derivative. You actually need the full upper convected derivative, which includes the material derivative as well as those two nonlinear terms, which come from stretching and rotation of a tensor. And then you represent the strain rate tensor as is typically done as the symmetric part of the velocity gradient. And so then you end up with this equation, which is the upper convective Maxwell model, which if you then couple it with an additional viscosity from the fluid, then you end up with a Stokes-Oldroyd B model. And as I've written it, we have possible parameters, dimensionless parameters. You can call it the Deborah number. You can think of the Weisenberg number instead, but basically either one of those parameters is proportional to the relaxation time. The Deborah number is defined if you have a characteristic flow time scale that you then normalize by. And the Deborah number is sort of the better parameter for swimming organisms. And then you also have another parameter coming from the ratio of the solvent to the total viscosity. And I like to think of that as fixed, because if you have a particular fluid, then that ratio is going to be fixed. So I'll pick a reasonable beta and just move on at this point. And so when you want to do modeling of a swimmer, then the question really becomes, what are you asking? And so in what follows, I'm going to try to tell you guys about a couple of different models where we assume different things. And with the different assumptions, you're asking different questions. And this is one of the beauties of math modeling, is if you design your system right, you can ask a particular question. But of course, it's way more complicated than that, and you never know what's going on in the beginning. So the rest of my talk is going to be organized in 3 lessons. And lessons I have learned over my, like career doing this kind of work, which has now been going on for a little while. About as long, I remember very distinctly because I started working with Bob the summer after my second child was born. So his age is kind of how long I've been in this game. So he's turning 13 next week. Been a little while. All right. So these 2 different examples come from different models. The 2D penalization method is what John was talking about. And then we also have models where we prescribe the shape. And so the body positions are fixed in the swimmer's frame, and we just put them into the fluid and watch them go. And so when I did jump into the game, this was kind of what was going on in the literature, where we were frequently measuring the swimming speed as a function of the Deborah number, and we were asking, if we put the swimmer in a viscoelastic fluid, does it swim faster or swim slower? So we would plot the normalized swimming speed, so the velocity divided by the velocity in a Newtonian fluid, and we would just ask the question, does it speed up, does it slow down? So that was kind of basic question back then. And there was a lot of really great work, including work by Lisa Fauci, where people were doing experiments, simulations, more experiments, biological experiments on the left. Thank you. Physical experiments on the right. And, people were getting different results, and people were puzzled by this because, as you see on the left, you have a few contradictory graphs. On the right, the green line on the left is actually that simulation from the middle. And then this one on the right came a little later, where they were actually seeing this sort of monotonic increase in swimming speed. And so lesson 1 was appearances can be deceiving, because we wanted to copy the Traum-Fauci-Shelley work in a book chapter, just as an example. And so we said, we're going to do what they did. And so we prescribed a traveling wave down the body that was just like what they did. And we thought we'd be cute. And we were like, but Paolo's writing another chapter in that book. We'll put some of his experimental data into our model. And so we looked, and we're like, it's the same swimmer. It's just like the Traum-Fauci-Shelley swimmer. But the traveling wave that we prescribed on the body was different if we prescribed it going right or going left. And in one case, the curvature amplitude was much larger in the tail, and on the other case, the curvature amplitude was much larger in the head. And it turned out that we got completely different swimming speed results, depending on which direction that wave was going. And in one case, we called it the kicker, where the head was on the right. We would match the Traum-Fauci-Shelley, but in the other case, which was matching the kinematics of the experiment better with the worms, we got a swimming speed that was decreasing. And so we could kind of recapture those results on the left, where we have both the non-monotonic increase as well as the decrease, just depending on the direction of the traveling wave. There's one other little detail that I'm not really going to go into. Well, I'll go into in a minute. But for us, we were looking at the shapes, and we're like, they're the same. So why would this be different? And it was a very surprising result, which actually turned into a paper as opposed to just a book chapter. It was amazing, but it was really cool. So body flexibility does also matter. And this is lesson 2. So the Traum-Fauci-Shelley paper, they showed non-monotonic speed up for these undulatory swimmers with a large tail amplitude. But this other group with the physical swimmers, they showed this, it's still non-monotonic, but if we ignore what happens, their low Deborah number, it's really a monotonic increase. And in particular, the increase is huge. So if you look at that U over UN, it's 2 on the right, whereas it's 1.3 on the left. We said, what causes this extra swimming speed boost? And it turned out the lesson is watch those parameters, because in the numerical experiment, you change your parameter, your Deborah number, by changing the relaxation time. I don't know, that just seemed more sensible than changing the period of oscillation, right? For us. But in the physical experiment, they actually changed the Deborah number by changing the period of oscillation. And I only learned about, this is another like cautionary tale / lesson, is talk to people and ask a lot of questions, because I only learned of that when I asked Roberto Zanet, who is the PI on that paper. I asked him about the experiments, and he told me that making a viscoelastic fluid was very hard. And so when you have your fluid, you like save it and you never touch it, and you don't want to make another one because it takes forever. And I was like, well, if you don't want to make another one, how do you change your Deborah number? Oh, you've just got a little motor that can change the speed of oscillation of your little physical swimmer. And he's like, yeah, of course, I just changed the motor, I just turn in the crank. That's the easy thing to do there. And yes, lo and behold, we all know that if you change a non-dimensional parameter 2 different ways and you get 2 different answers, then you're missing something, right? So watch those parameters. And in this case, the missing thing was actually that bend. It's like a little subtle, but it was that bending stiffness in the model where we prescribed the curvature, and different shapes were coming. And so we could actually match the Zanet result if instead of varying the relaxation time, we varied the period for sufficiently soft shapes. And so the picture is the black line there is my target shape. So that's the goal. But my blue and my red lines are the result of swimming in the model. And what you'll notice if you stare at those videos long enough is that the red swimmer, which is the swimmer in the viscoelastic fluid, sort of hugs that target shape a little bit better, right? Its amplitude is slightly boosted. And we could actually prove that with asymptotics, which was super cool. And so that was the end of that story. And the final story starts with clammy, and I'll just show you briefly the work that we did where we prescribed the shape of the clammy. And so what we were after was trying to understand swimming speed results that Paolo's group had for the clammy swimmer as a function of the Deborah number. And they were seeing this decrease in swimming speed based on these shapes. And so we said, all right, let's put ours in a viscoelastic fluid. Let's see if we can match the swimming speed. So we had this cool model. We got this decrease and we matched the experiments and we were like, yay, hooray. But this was a little while ago. And of course, one wants to keep doing work and one asks these questions like what's missing? What did we actually get? What did we not get? And one thing we didn't get was we didn't get anything about the frequency because our model prescribed the shape. So it's going to prescribe the frequency. So in their paper, in their experiments, they saw that the frequency increases with the fluid elasticity. So here's lesson 3 for everyone. Modeling is hard, but experiments are harder. And I have just been like floored by that with stuff I've been spending a lot of time with lately, which is I'm not going to show today necessarily, but the variability, right? The one thing that like modeling is hard. You have a lot of questions you have to ask. You have a lot of questions you have to answer, but the variability that you obtain in experimental data is so high, and trying to trust it and understand it, that's something I've been like, I keep asking Paolo and he's like, haha, welcome to my world. So here's a picture that sort of shows you some of that. And here we're going to use swimming speed as a method to quantify stroke variation. So here's where the regularized Stokeslets come in, because we're just going to look at the swimming speed in a simple regularized Stokeslets model to ask the question of like, what happens when you change the shapes? And so this work we have published, it's going to appear later in September. But so we obtained, this was data Paolo gave us, and each one of these images comes from a different experiment. There's already some averaging having been done to obtain these shapes, but these are the different shapes. And so you'll notice on the left there, you've got 3 different viscosity 1 shapes. Then you've got increasing viscosity, and then I think there's one other repeat. There's a 2 at 3.45, and then there's 2 at 6. But there's a lot of variability in sort of how to quantify that and how to even like understand what's going on is a really hard question. So we said, all right, well, let's first let's ask the question, how much does the shape matter? How much does the frequency matter, right? Because I already told you the frequency goes down. So if the swimming speed goes down and the frequency goes down, maybe they go down together, but no. So the speed as a function of viscosity goes down by from 150 down to like 30. The frequency versus viscosity decreases by about half. Right, so that does not, explain the speed versus viscosity graph. And so if you actually just measure net displacement versus viscosity, so forget frequency, so rescaling, you still get a decrease. So something about the shapes is actually changing the swimming speed. So sorry, that was experimental data that I was just showing you. Okay, so we wanted to model the strokes, and we're asking, how do we model the strokes? And so what we did was we used this idea that we found in this paper from 2014, where they were modeling both sperm and flatworms, and they were doing like a mode decomposition. So simply just a low mode decomposition. So nothing mathematically not super novel, but very complicated and really interesting to apply to shapes. Okay, and so you obtain a mean tangent angle, and then you look at these shape modes, and you can see in this example that comes from their paper that the two shape modes explain nearly all of the dynamics in the motion. So they say, okay, well, I'm going to use this shape model where I have a mean tangent angle, and then I have my two shape modes, and go on with my life. So we looked at clammy, and it was not that straightforward. So we are going to apply this to the clammy data, and we had 9 or 10 different data sets in something like 5 different fluids. I forget exactly. This was the variation in the mean shape. And the color bar there represents the viscosity. So all the sort of similarly colored flagella there are coming from a similar viscosity, and you'll see that it's kind of non-monotonic, it's kind of confusing, it's a little bit all over the place, and this is just the mean shape, right? So then what happens when you look at the shape modes? And so we decided to take, I mean, we looked at a lot of different things, but we decided to take all the different shapes together and make a giant shape basis. So we created the shape basis, and we were able to see that, with I don't know, 2 modes, we had about 92% of the data. By the time we got to 4 modes, we got to 99.1 or something. And so these were the shape modes that came out. And then the question is, what are the different experiments? So for the different experiments, we project onto these shape bases, and we get these different temporal frequencies, which don't look like single frequency data, right? So, 1 frequency, 2 frequencies. She looked at the different coefficients for like up to 4 frequencies to see like what kind of model do we want to use for our thing. Because basically the point is that we didn't want just a simple 2 shape mode, 1 time frequency model. It seemed like the data was more complicated than that. And so here's an example of the shape reconstruction where on the top, it's 2 modes. On the bottom, it's 4 modes. And then it's increasing temporal frequency. And I don't know, if you stare at this, like the question is, it's a really hard question. Like, well, what's the best, right? Like, I don't know. I mean, how do you really pick? So what I think that the key idea was using the swimming speed as kind of a metric and saying, all right, well, how much does the swimming speed change in all these different examples when you use the different types of models, right? So here's a graph on the left, which shows the difference within the experiments, comparing to kind of like pretending that the true model comes from 6 shape modes and 4 frequencies. I mean, we could have done higher, but that seemed like fine. And you see that the variation in the speed difference is much more significant with fewer modes. And when by the time you get down to 4 modes and 4 shape modes and 2 frequencies, you're kind of pretty minimal differences. I mean, you can go higher, but we decided to stop. So that was what we decided. And I know I'm running out of time, but I do want to show you the great clammy race. I mean, who wouldn't want to see? So on the left is our swimmer. This is, so from now on, what I'm going to show you is 4 modes, 2 frequencies. And so like the question is, which one's going to swim faster? If you've been paying attention, you know the answer. Well, unless our model's really bad, right? So you can see that the shapes really are quite different, right? If you look at them, and that the viscosity 1 is going faster, right? I think when you're watching this, it's easy to sort of think initially this one might be faster, but then the other guy speeds up. So how does the model compare to the data? Pretty good. So the data's in red, the model's in blue. Great. And so the model captures the correct trends. So you could stop there. You're like, yeah, our model works. But we were kind of curious, like, what matters, right? Like, how much do these different parts matter? And right, like, there was some kind of, we had to make choices at some point. And so we asked this question, how important are the different components of the stroke? And so, real quick, I'm just going to show you sort of how this decomposes. We have this, because these are not like, the mean's not zero, right? This is like already curved. And so when the mean is from a low viscosity shape, the mean comes down a little closer to the body, whereas these, they sort of stay away from the body. Why? That's a big question that we still don't know the answer to. But what you can do is you can actually make these kind of Frankenstein swimmers. And so in this example, the stroke on the left is coming from a low viscosity. The stroke on the right is coming from a high viscosity, but the stroke in the middle is a high viscosity mean shape with a low viscosity stroke on top of it. And so what do you notice? You notice that that mean shape is really keeping the flagella away from the body, right? And so now you can say, like, who wins when you throw in, I didn't put Frankenstein on the slide, but who wins when you throw in the hybrid? And, I guess I could stop and make you guess, but I am running late and I don't want to interrupt your coffee too much. So, yeah, it's still that viscosity one, right? It's still the original low viscosity swimmer that wins. So we were surprised by this because, when you think about it, you think the stroke is what matters. But no, it's not the stroke. The stroke doesn't matter. It's the mean shape that matters. And so this is a messy slide that's a little hard to digest. So I'll just tell you the punchline is, where do the dots line up better with the trend lines? Not on the left, on the right. Here, where what we're doing is the stroke is fixed. So the actual shapes are according to, are fixed. They're all the same. I don't remember which stroke. But the mean changes according to the experiments. And so the mean shape, and then this is sort of my last like real slide, the speed dependence on the shapes comes from the changes in the mean. And so if you compare the speed with fixed stroke, you get this, they're all a little bit different, but the scale of that variability is actually on the same size as the scale of the cell-to-cell variability we already have. So it's really important that the mean shapes are changing. And that's kind of nice because I like simpler things, right? The mean shape's a lot easier to deal with than the stroke. So maybe we really just need to ask why is the mean shape changing? Yeah, that's just a summary. And so what causes changes to the mean shape? How does this story change with viscoelasticity? These shapes arise from this complex coupling. We know there's the motors are involved, et cetera, et cetera. And then, of course, what about other organisms? So, there's so much more to do. It's a really cool project to be working on. I'm really grateful to get to study this stuff. And thank you for your time.
Thanks for the lessons. I was just wondering if you would comment on like, two different observations. In the beginning of your slides, when you're showing the chemographs, we saw with viscosity increasing, the frequency went up.
Yeah.
And in the end, when the Calmi ones, we saw as viscosity increasing, the frequency went down. Is there any way to reconcile between these two behaviors?
It's a good question. I don't know. I think the thing to do is really to study the models and understand what's going on. Viscoelasticity does seem to increase frequency, and we did do a paper on this with a very simple model where we drove a filament with like a follower force at the tip. So we have a filament and we just put a little driving force right at the tip. So the follower model, if you're familiar with it, and we put that in a viscoelastic fluid, and we did see that like viscoelasticity increases the frequency. And we're actually able to match that to an asymptotic model as well. So there is something about it that increases frequency, but viscosity decreases frequency. So then they're competing effects. And so when does what win?
So you said early on that it's really hard to change the viscosity, but then with the clammy results, you said you had 5 different viscosities.
Yeah, so it's a different experimental.
And that was also in the experiments. So was it one person doing the experiment and changing the solutions, or like 5 completely different?
No, that was one person change. Yeah, so Paolo.
How did they change the viscosity?
Well, like
It's so hard.
So Paolo is the expert in viscoelastic fluids, and he talks about how hard it is, but he's done lots and lots. I mean, it's not impossible. So it really, it's a question of like, what is your experimental lab usually doing? And are you an expert in this or not? And I think the key answer is like, you can't just jump in and say, oh, I'm going to make viscoelastic fluids, right? Not everybody can do that. And you have to put a lot of time into it and develop the material. So yes, it's hard, but it's not impossible.
A question regarding organisms and how they interact with the mucus. You're looking primarily at physical phenomena associated with a swimmer that's pushing through the mucus. Is there interest in looking at chemical relationships? How the swimmer modifies the viscoelasticity? Do we even know exactly about that? I mean, a lot of swimmers may release enzymes that interact with the mucus so that they can swim through it, but other swimmers can't. Do you know people that are looking at that either from experimental or modeling standpoint?
Yes, absolutely. I mean, so there's sort of 2 questions there and many questions there, but when it comes to like it's not just the fluid and the swimmer, obviously, and then it's also not just the motors, because chemistry is an important part of it. And I know like Lisa and Sarah have done work on like signaling. So there's definitely chemical sensing and whatnot. So in terms of actual motion, like why would a swimmer go a certain way? It's not just random a lot of the times. But in terms of like thinking about modifying the fluid itself, yes, there are also people who've looked at that. And I know, I mean, some of this work can be a little bit controversial if you get really down in the weeds, especially depending on the scale you're at. Because I know that when you're at like prokaryotic scale sizes and things like that, like when you're so tiny, the fluid doesn't necessarily look like a continuum. And so some of the arguments that we make based on like experiments we do with macroscopic fluids might not even be relevant. And there have been work where people look at kind of thinking about a capsule of like low viscosity fluid surrounding an organism as it's making its way through a mucus environment and things like that. I'm not familiar with exactly how that happens, but I know people look at that. So absolutely. Yeah. So modeling is also hard, right? All of this is hard. There's a lot going on. Yeah.
Okay, let's thank our speaker again.